Good morning to all my dear students. Myself Jameel Ahmed, Assistant Professor in the College of Pharmacy. Today we are on lecture Pharmacognosy and Phytochemistry. Unit first, chapter first, under the subject metabolic pathways in higher plants and their determination. Under this chapter unit, we have to cover 2 points. A brief study of basic metabolic pathways and formation of different secondary metabolites through these pathways. Under this heading, we have to cover some metabolic pathways like shikimic acid pathway, acetate pathway, and amino acid pathway. These 3 pathways we have to cover in your syllabus. We will cover it one by one in our lecture series. So the contents of our today's lecture, which is related with the metabolic pathways. To cover this, first we should know the metabolism. This word is not new, as you have heard many times in your previous years in biochemistry and anatomy. So, what is metabolism? Metabolism is nothing but it's carried out in 2 processes. Have you remember what is that? That is anabolism and catabolism. Now, what is anabolism? Anabolism is the building blocks of the elements in our body. You know catabolism? Catabolism is the breakdown of the foods. Whatever we are eating, it gets breakdown and then going to the circulation, from which we get the energy. Now the question is, where is this process occur? We know that this process occur in our liver. That is the most important vital organ. In this process, various biochemical reactions carried out for this enzyme. For this, enzymes plays an essential role. These all information is for your basic metabolism, which happens in our body. Same thing is happen in plants also. In our pharmacognosy, we are discussing the metabolism in plants. Now come to the definition of metabolism. Metabolism can be defined as sum of all biochemical reactions carried out by an organism. There are 2 types of metabolites are there. Metabolites. What is those? Number 1, primary metabolites, and second one is secondary metabolites. First we should know the metabolites. In biochemistry, these are the intermediates or end products of metabolism. Means metabolites are the intermediates or end products of metabolism. The term metabolite is usually used for small molecules only. Metabolites have various functions, including fuel, structure, signaling, stimulatory functions, and inhibitory effects on enzymes also. The catalytic activity of their own, usually as a cofactor to an enzyme. You know the enzymes, role of enzymes is very important in metabolism. The same thing is happen here also. The enzymes cofactors are the essential roles in metabolites also. Metabolites have different functions, they mean as defense also, and interactions with other organisms, example pigments, odorants. So these are the functions of metabolites. Now come to the primary metabolites. You have to know about the primary metabolite. Primary metabolite is directly involved in normal growth, development and reproduction. You must understand that primary metabolite is directly involved on normal growth of the plant and development of the plant and they involved in the reproduction of the plant itself. If you can see the example, ethylene exemplifies a primary metabolite produced large scale by industrial microbiology. This one is about primary metabolites. Now a secondary metabolite, have to know, that is very important in your syllabus. A secondary metabolite is not directly involved in those processes but usually has an important ecological function. These are not directly involved in those processes, just like primary metabolites, but usually it is an important and ecological function. Example, antibiotics. We know the antibiotics. Antibiotics and pigments such as resins and terpenes, etc. These are the example of secondary metabolites. Some antibiotics use primary metabolites as precursors, such as actinomycin, which is created from the primary metabolites tryptophan. Means actinomycin is created with the help of primary metabolites to form a tryptophan. Second one is some sugars. Sugars are the metabolites such as fructose or glucose, which are both present in the metabolic pathways. We know that as our previous studies. Glucose is an important parameter in the metabolic cycles. To understand this, we have to get the difference between primary metabolites and secondary metabolites. Primary metabolites are widely distributed in nature. Practically in all organisms. And to go to the secondary metabolites, which are biosynthetically derived from primary metabolites, more limited in distribution. These are restricted to taxonomic group only. Secondary metabolites. Now the second difference in primary metabolites, they have directly involved in growth, development and reproduction. In secondary metabolite, they are not directly involved in growth and development. they are involved in chemical adaptation to environmental stress. They serve as a defensive, protective or offensive chemical against microorganisms, insect and higher herbivorous animals. This is the most important difference about secondary metabolites. Now, the third primary metabolite difference, generally they are non-expensive to produce, to isolate from plant or plant parts. Example, starch. Starch we can get from potatoes easily. In secondary metabolites, they are generally expensive to produce. Example, quinine. Quinine is isolated from cinchona bark. Quinine, we know the uses of quinine. Quinine is an anti-malarial drug. Nowadays, chloroquine is a very demandable product in this pandemic situation, COVID-19. So we get this small quantity, and this is the expensive process. Now the primary metabolites, they are present in plants in large quantity. Secondary metabolites present in plants in much smaller quantity. Primary metabolites are simple in nature. Secondary metabolites are complex in nature. Naturally they are complex. They are structurally complex in structure. In primary metabolites, generally do not have any biological or pharmacological action. Therapeutic value, regarding the therapeutic value. Secondary metabolites have marked pharmacological action. They have therapeutic activity for human body. They are responsible for medicinal value of plants also. The examples of primary metabolites: carbohydrates, starch. Under this carbohydrate examples: starch, cellulose, sugar. Lipids, fatty acids. We can get fatty acids from lipids. Proteins. Proteins are nothing but the amino acids. And nucleic acids, we know that the RNA, DNA, these are nucleic acid. This is important primary metabolites. Now the examples of secondary metabolites: alkaloids. Alkaloids, you know that alkaloids are the basic metabolism, basic in nature. They are present in as a secondary metabolites in plants. Glycosides are there, flavonoids are there, resins are there, terpenoids are there. These are the difference between primary and secondary metabolites are there. See here the schematic diagram representing integration of primary and secondary metabolites. Here the primary metabolic pathway of carbon. There are so many cycles are there. Krebs cycles, as you know, the glycolysis cycle. As you studied in your biochemistry, these are the cycles. From these, the primary metabolic products we can get, and from primary metabolic products we get the secondary metabolites. Now come to the classification. classification of secondary metabolites. There are 5 main classes are there. Number 1, terpenoids and steroids. Second one, alkaloids, fatty acid derived substances and polyketides. The fourth one, non-ribosomal polypeptides and enzyme cofactors that plays an essential role. This classification, as you heard in the screen, over 21,40,000 secondary metabolites are known. These are commonly classified according to their vast diversity in structure, regarding their function and their biosynthesis also. So among these, 5 are the main classes. These are the terpenoids. Under these terpenoids and steroids, they are a major group of substances derived biosynthetically from isopentyl diphosphate. Currently, 35,000 known terpenoid and steroid compounds are identified up till now. Amongst these, terpenoids have different variety of unrelated structures. Steroids have a common tetracyclic carbon skeleton and are modified terpenoids that are biosynthesized from the triterpene lanosterol. Structurally, steroids have a basic steroidal skeleton, 5-membered tetracyclic carbon skeleton is there in their structure. Alkaloids, second one is, alkaloids are the basic nitrogenous amino group compounds which are derived biosynthetically from amino acids. Fatty acid derived substances and polyketides. Around 10,000 compounds are identified and are biosynthesized from simple acetyl precursors such as propionyl coenzyme, acetyl coenzyme and methyl malonyl CoA. Fourth one, non-ribosomal polypeptides. These amino acids derived compounds and are biologically synthesized by a multifunctional enzyme complex without direct RNA transcription. Non-ribosomal polypeptides, these are related to a nucleic acid. You know that nucleic acid synthesis. Fifth one, the important is enzyme cofactor. Enzyme cofactors are non-proteinous in nature. They have low molecular enzyme components. They are very essential role in metabolic pathways. Now, the second point is we have to discuss here the functions of secondary metabolites. The major function of the secondary metabolites, including, as I have discussed earlier in this topic, antibiotics. Antibiotics, you know that antibiotics are the products which are used to kill the bacteria or microorganisms. Antibiotics are prepared from microorganisms, we know that. They have an essential role on their competitive weapons against other livings such as animals, plants, insects and microorganisms. Then metal transporting agents, antibiotics are the, or secondary metabolites are the metal transporting agents. The secondary metabolites are the agents for symbiotic relation with other organisms also. They are play essential role in the reproductive agent as a and differentiation factors. So we can differentiate from each other with this help of reproduction. Means X chromosomes, Y chromosomes can differentiate. Now, the functions, agents of communication between organisms also. The other functions include the spore formation. They play as an essential role in spore formation, not obligatory, but in germination. The secondary metabolites are used for variety of biological activities. Antimicrobial is the agents which kill the microorganism or it effective against microorganisms, and anti-parasitic agents, the agents which are used to kill the parasitic organisms, and in enzyme inhibitors, and it acts as an anti-tumor agent also. Anti-tumor agents is cancerous cells, which treat the cancerous cells. Immunosuppressive agents also. These all are the functions of secondary metabolites. Now, last, we have to know the sources of secondary metabolites. They are 80% of secondary metabolites sources comes from plants. Remaining comes from bacteria, fungi, and many marine organisms, that is sponges, tunicates, corals, and snails. So, my dear students, this is the introduction of basic secondary metabolites. We will discuss in our second lecture the metabolic pathways. Thank you very much.